Macrophages acute inflammation. Via the toll-like receptor pathway, you get the production of the cytokine IFN gamma, which can lead to the activation of macrophages. Once activated, they can contribute to the phagocytosing of microbes. A microbe has a PAMP, which binds to a PRR receptor found on a macrophage, which results in the triggering of phagocytosis. Macrophages also help with the activation of endothelial cells, which is one of the vascular changes made by the body to help contain an infection. In response to damage, macrophages within our tissue produce IL-1 beta and TNF alpha, which binds to receptors on endothelial cells and gets you the upregulation of E-selectin and ICAM1 and VCAM. Upon binding, chemokines are also released, which stimulate chemotaxis of macrophages. Macrophages also help with efferocytosis. After a neutrophil has cleaned up a microbe, it undergoes apoptosis and is removed by macrophages. IL-10 and TGF-beta are byproducts of this. Macrophages wound healing. During the proliferative phase of wound healing, macrophages move into the wound space to help formulate granulation tissue. During the tissue remodeling phase, macrophages undergo apoptosis, and activated macrophages secrete growth factors, which helps with the fibroblast proliferation and chemotaxis of more macrophages. Macrophages chronic inflammation. Granulomatous inflammation contains granulomas, which have aggregated macrophages surrounded by various T cells. Additionally, macrophages are heavily present in non-granulomatous inflammation. The main cause of this inflammation are when an irritant fails to be eliminated and when self-antigens formulate an autoimmune reaction. Also, once a macrophage is activated, it releases IL-1 and TNF alpha and presents as an antigen to a lymphocyte. This triggers the activation of a lymphocyte, and IFN gamma is released. IFN gamma then goes on to activate more macrophages. This process then repeats. With lots of macrophages, we get constant non-specific tissue damage via the release of reactive oxygen species and reactive nitrogen intermediates. In response to chronic inflammation regarding autoimmunity, molecular mimicry takes place. A pathogen often has something that resembles our self-antigens, which triggers a self-reaction response. An antigen-presenting cell would then present the pathogen's antigen, which is recognized by our helper T cells, leading to the activation of macrophages. These activated macrophages then cause non-specific damage. Macrophages process the antigen involved in delayed type hypersensitivity reactions as well by stimulating, or which stimulates Th1 cells. More macrophages are then recruited to the antigen site by chemokines and IFN gamma. Neutrophils acute inflammation. Additionally, the liver produces fibrinogen, which is cleaved into fibrin and fibrinopeptides by thrombin. Fibrinopeptides are chemotactic for neutrophils. Neutrophils also play a role in the activation of endothelial cells. With the upregulation of E-selectin, neutrophils can bind via its mucin receptors. This slows down neutrophils. Also, thanks to the cytokines macrophages produce, chemokines are secreted. These chemokines are able to bind to GPCRs, which further activates neutrophils. These cytokines produced by macrophages also cause upregulation of VCAM and ICAM1, which helps adhere neutrophils. Neutrophils aid in resolution. With phagocytosis, the neutrophil engulfs its microbe. Regarding degranulation, the neutrophil has these granules with enzymes. These granules merge with the neutrophil's plasma membrane, allowing for the release of these enzymes, which degrade the microbe. In netosis, the neutrophils can extend net to their DNA to trap the microbe. Within the net are enzymes such as elastase, which help break down the microbe. Neutrophils wound healing. Neutrophils are heavily involved in the inflammation phase of wound healing. Neutrophils work together with the macrophages in order to clear the site of debris, as previously mentioned, via netosis, granulation, and phagocytosis. Neutrophils chronic inflammation. Neutrophils are also heavily present in other even non-granulomatous inflammation. Regarding autoimmunity, neutrophils cause cell-mediated damage. Neutrophils, along with macrophages, release cytokines and chemokines, which allows for the release of cytotoxic cytokines, reactive oxygen species, and nitrogen intermediates. Regarding autoinflammatory conditions, there is a substantial increase of neutrophils, which leads to increased release of IL-1, which can be very harmful. Overall, macrophages and neutrophils play a vital role in acute and chronic inflammation as well as wound healing.